Hello, everyone. My name is Ai-Ling Lin. I am professor and vice chair for research in radiology. Today, I'm going to tell you about how we translate our animal studies into a clinical trial. And specifically, we are using sirolimus, or known as rapamycin, to potentially mitigate Alzheimer's disease risk, especially for those who are genetically at high risk. So, what is Alzheimer's disease? It is a neurodegenerative disorder and also the most common form of dementia in elderly. There are three major hallmarks: ATN. A is amyloid beta plaques outside the neurons, and T is tau tangle inside the neuron, and N is neurodegeneration. Alzheimer's disease patients, you can see that has a brain volume shrinkage compared to the healthy individuals. And now with advanced imaging technologies such as MRI and PET, we can see this disease progression in action. MRI can tell us the detailed changes in brain structural shrinkage, and also FDG PET can tell us the glucose deficit or hypometabolism in the patient compared to healthy individuals. So there's a genetic risk factor for late onset Alzheimer's disease known as the ApoE4 gene, apolipoprotein E4 allele. This apolipoprotein is a gene regulating lipid metabolism, and there are three variants. E2 is protective against Alzheimer's disease, E3 is neutral risk, and E4 increase the risk. And you can see that just because the amino acid difference from cysteine to arginine that made the protein structure very different for the E4 carriers, and therefore the people with this gene have higher potential to bind with the very low density lipoprotein, which is known as VLDL, and that is easier to lump with the amyloid beta plaques. So the individuals who have one or two ApoE4 copies will have higher amyloid beta retention, and that lead to a 4 to 8-fold increased risk for developing Alzheimer's disease, and the age onset is also reduced to 7 to 15 years compared with non-carriers. And before having A beta plaques or tau tangles, even decades before the cognitive impairment occurs, cognitively normal ApoE4 carriers already show brain hypometabolism from the FDG images. You can see that young adults or middle-aged adults, their brain already show hypometabolism in the regions showing in light blue here, and the purple ones are the regions from Alzheimer's disease patients. So you can see there's already a brain region overlapping in terms of hypometabolism between healthy adults and Alzheimer's disease patients, and those brain regions increases as time goes. In addition to metabolic deficits, ApoE4 carriers also have early vascular dysfunction, such as blood-brain barrier breakdown, showing in panel A. That is because of increased inflammation in the vasculature. Panel B shows the reduced cerebral blood flow in young adults. Cerebral blood flow is very critical because it supplies the nutrient to the brain region. So with the reduced CBF, this also aligns with the finding of hypometabolism because glucose and oxygen supply goes down. Panel C shows the functional MRI outcomes. This is a memory task, and from the picture we can see that ApoE4 carriers, they need larger brain region to perform the same task compared to non-carriers. This indicates that their brain may need to work harder to get the same outcome. And the finding also align with the hypometabolism or lower perfusion because the brain has less energy supply. In our lab, we have been using an animal model that can mimic human condition in terms of Alzheimer's disease development and cognitive impairment timeline. So using this EFAD mice, we also showing that they have cerebral blood flow reduction way early before they have amyloid beta plaques, and that is also earlier, way before they show cognitive decline. So between 2 to 6 months, we thought that's a window opportunity to come in to intervene to potentially mitigate the risk for Alzheimer's disease pathology development or even preventing the onset of cognitive impairment. So we focus on a signaling pathway called mTOR, mammalian target of rapamycin. mTOR is a nutrient sensor. So if when the nutrient is abundant, then we can see that the mTOR will be activated to promote growth or cell proliferation. But when this pathway is inhibited, it shifts the metabolism into maintenance, such as cleaning DNA damage or clear up the misfolded proteins, such as amyloid beta. And then rapamycin is an mTOR inhibitor. It has been FDA approved and has been using to treat other diseases. And we found that by inhibit mTOR, actually it can be very protective for maintaining vascular and metabolic function. So in our study, we're just thinking about if we give rapamycin early enough, between 3 months to 7 months before the mice have a severe A beta plaques or cognitive impairment, can we restore their brain vascular and metabolic function? And using MRI, we can do a pre and post measurement. From panel A, you can see the before treatment. The ApoE4, E4 fat mice already have significant reduction in cerebral blood flow. And in panel B, you can see that after the treatment, the E4 mice with rapamycin actually have a restored cerebral blood flow compared to the control group. And that level, the restored CBF level actually is very similar to that of E3 mice. And we do a pre and post CBF calculation, and you can see that E4 mice have significant increase in their cerebral blood flow, especially in females. This is significant because females have higher risk incidence for developing Alzheimer's disease compared to males. And we found that the pathway could be because mTOR can actually activate endothelial nitric oxide pathway, and which will increase nitric oxide release. And nitric oxide is a vasodilator, so that could potentially also increase cerebral blood flow. And we also look into blood-brain barrier function, look into P-glycoprotein, PGP, which is a transporter on blood-brain barrier to clear amyloid beta from the brain tissue into the vessel. Panel A and B shows the microscope findings, very similar to CBF findings. E4 mice with rapamycin has restored PGP level, and to the level very similar to E3 mice. Panel C shows the Western blot findings, very consistent with the microscopic image showing that rapamycin increase the PGP level for E4 mice. We also do A beta staining, and you can see that with rapamycin, the brown dots in the E4 fat mice brain has been significantly reduced, which means A beta has been successfully clear out from their brain. And using MR spectroscopy, we can measure brain metabolites. Panel A shows the voxel placement on the hippocampus of the mice. Panel B shows the corresponding spectra from that voxel. Panel C to E shows the metabolite changes from the treatment group. You can see that E4 mice with rapamycin has restored level compared to the control group. So phosphorus calling on panel C shows the cell membrane integrity. Panel D, the glutamine, they are the neurotransmitters. They are also restored by rapamycin for the E4 mice. NAA in panel D is N-acetylcholine aspartate. It's a neural integrity marker, and you can see that it also being restored by rapamycin for the E4 group. With all these findings, very exciting, and with rapamycin as a FDA-approved drug, we thought we may translate our findings into a clinical trial. So this time we're also looking to cognitively normal middle-aged participants, because that's the window opportunity to potentially help them preventing getting Alzheimer's disease. So this is the inclusion and inclusion criteria. We recruit cognitive normal participants. We will stratify their ApoE genotype and to do 6 visits. So visit 1 is eligibility visit to take an oral swab to measure their ApoE status, and also cognitive testing to make sure they pass through the standard of cognitive function. And visit 2, we will do a baseline MRI and also take their blood sample to be able to know their inflammatory marker changes, as well as the Sirolimus level in their blood. So in clinical trial, we call it Sirolimus instead of rapamycin. It's because the ingredient is a little bit different for giving to mice compared to humans. But they are the same, potentially the same compound. And in visit 3, we will give them the medication. And every 2 weeks, we will get them back to do MRI scan. Until visit 6, we'll take another blood sample and to do the final measurement. So in this study, we're using the most advanced MRI technology scanners in NextGen. We have 130 MRI and 70 MRI. This 70 MRI is currently the only one in Missouri. These 2 machines allowed us to do the high resolution and very advanced measurement for structural scan and cerebral blood scan. And also 70 give us a very detailed resolution of spectroscopy compared to 3 Tesla. And with very early preliminary data from first subject, we see very encouraging outcome. Only within 2 weeks of giving Sirolimus, this participant with the ApoE4 gene have a 20% increase in their cerebral blood flow. And this increase has maintained throughout the study. So this is very encouraging and very fit our hypothesis that rapamycin can restore vascular function. In this study, we also further look into gut-brain axis, because the field start to know that the amyloid beta plaque and tau tangle may not just coming from the brain itself. It could come from other organs, such as the imbalance of bacteria in our gut. This is called dysbiosis, indicating the imbalance of harmful and beneficial bacteria in our GI system. So if our gut has more harmful bacteria, it will produce inflammatory cytokines and change the composition of microbiota in our gut system. They can lead to increase in pro-inflammatory cytokine production, or more harmful metabolites, and that lead to inflammation, lead to microglia activation, and down the road lead to blood-brain barrier breakdown and amyloid beta plaques formation, tau tangle formation. So in this clinical trial, we also collect their stool sample and sequencing the bacteria composition from the samples. finally, we will correlate the microbiome findings with the brain imaging marker. So we will welcome you to come to observe our clinical trials. We will continue this trial maybe till the end of summer. So summary from this presentation is that I hope you know that ApoE4 is the strongest genetic risk factor for late onset Alzheimer's disease. If ApoE4 carriers have reduced cerebral blood flow and brain metabolism earlier than non-carriers. Rapamycin may mitigate Alzheimer's disease risk for asymptomatic ApoE4 mice by protecting their brain vascular and metabolic functions and increasing A-beta clearance. And we also find, actually, from the animal study, is that the response to the drug is ApoE genotype dependent, also sex dependent. I don't have the time to show you the data today, but ApoE3 mice respond to the drug very differently. So we thought that pharmacogenetic effects is the effect responding depending on genotype will need to be considered in the future. And clinical trials are needed to verify the efficacy of Sirolimus or rapamycin in humans. And pilot study from our lab has shown the promise of cerebral blood flow restoration for ApoE4 carriers within 4 weeks of treatment. And on our website, we have a YouTube video to give you an introductory of this study from animal findings to clinical trial. And also here are some tips for you to know how to reduce the risk for getting dementia. More information can be found on our website, where you can email me for questions. Thank you very much for your attention.